Hello learners, welcome to Ahmad Coaching and I'm your teacher Dr. Anam. Today we will study about what is polymerase chain reaction. Polymerase chain reaction was developed by Karim Ollis in 1983. For short, polymerase chain reaction is known as PCR. So normally when you talk about polymerase chain reaction, you always say that we are doing PCR. In the laboratories, the people always use a short form, that is PCR. PCR is a method widely used to rapidly make millions to billions copies of a specific DNA sample. So this is helpful because it is going to help you to make millions and billions copies of a single DNA molecule. It can amplify a single gene or a small piece of DNA among all the genes of an organism. For example, if we take an example of the humans have 20,000 to 25,000 genes. And if you perform a PCR for a particular gene, you can even amplify a single gene out of these 20 to 25,000 genes. For example, I wanted to study leptin gene. Leptin is a hormone which is produced inside humans. So if I wanted to study its gene, I can amplify by the help of PCR and I can study that. To perform PCR, you need some specific components, and without the components, you cannot perform the PCR. So the components of PCR, first of all, we should have DNA sample. The DNA sample, it will contain the DNA target region to amplify. For example, if I wanted to study a specific gene in the human cells, what I am going to do that I'm going to take some human cells. I can take hair cells, I can take blood cells, I can take muscle cells, whatever the gene I wanted to study, I can take the sample accordingly. And from the sample, I'm going to take my DNA sample. So here we have our DNA sample. After that, I need a DNA polymerase. As the name indicates that it has A's, so it is an enzyme which is a thermostable enzyme. The thermostable enzyme means that it can withstand high temperature, which is used during the process of PCR. This enzyme is responsible for the DNA replication. The DNA replication is a process in which one DNA is converted into two DNA, or you can say that this is a process of DNA synthesis. Normally, the DNA polymerase is isolated from Thermus aquaticus. Thermus aquaticus is a thermophile, or it is a bacteria which can withstand high temperature. So scientists, they have isolated the DNA polymerase from this bacteria, and this DNA polymerase is sometimes also known as Taq polymerase. The Taq word coming from here. So here we can see that we have this Taq polymerase. The third component is DNA primers. The primers, they are actually short nucleotide sequences which are complementary to the 3' end of each of the sense and antisense strand of DNA target. For example, you wanted to amplify a particular segment of the DNA. So what you are going to do, you are going to design the primers which are going to be complementary to your DNA target. For example, my DNA target sequence is C, T, A, G, A. For example, so I'm going to make primer which is complementary to this sequence, which will be G, A, T, C, and T. So this is my primer. The primer should be complementary to your DNA target. Okay. We also call these primers specific primers because they are going to bind with the specific sequence of the DNA of your desired gene. They are not going to bind with any other gene, but they will only bind with the gene you wanted to study. So here we have the primers. There are two primers, the forward primer and the reverse primer. The one which will move from this side and the one from this side. So forward primer and the reverse primer. The fourth component that we require for the PCR reaction is deoxynucleoside triphosphate, or in short DNTPs. The DNTPs, they are actually the building blocks from which the DNA polymerase is going to synthesize a new DNA strand. We know that DNA is made up of nucleotides. So to make new copy of DNA, we also need nucleotides. So we are going to add our DNTPs, which are nucleotides, into the reaction mixture, and the DNA polymerase is going to use them to synthesize new DNA strand. So here we have our nucleotides. We have 4 nucleotides: adenine, guanine, cytosine, and thymine. And the fifth one which we require is the buffer solution. Buffer solution is going to provide a suitable chemical environment for the optimum activity and stability of the DNA polymerase. For example, if we are not providing the buffer, all these things are present but the buffer is absent, the reaction will not take place, because we have to provide all these things a suitable chemical environment so that they can work. So in the PCR reaction or PCR tube, we will add our mix buffer. So now we have added all these things in our this tube. We will place this tube into this thermocycler. This thermocycler is going to control the temperature which is required for the polymerase. Inside this, what will happen, the first step will take place, which is known as denaturing. The denaturing step takes place at 95 degrees Celsius. That is why we require thermostable enzymes, because 95 temperature is a high temperature. At this temperature, what will happen, that this DNA, which is a double-stranded DNA, is going to be separated from each other, or they are going to denature. So the both strands, they are separated from each other. Then the second step will come, which is known as annealing step. The annealing step will take place at 55 degrees Celsius. In this step, the primers, which are DNA primers, they are going to bind with each strand. The forward primer here and the reverse primer here, they are going to bind with the complementary sequence of the DNA, like I told you here. Okay, so this is the step of annealing. Annealing means that the primers, they are going to bind with the DNA strand. After that, the step 3 comes, which is the synthesizing step, and this takes place at 72 degrees Celsius. In this step, the DNA replication will take place, and the DNA replication will take place when the nucleotides will add into the strand. So here we can see is this stack polymerase is going to bind here with the primer and the single strand of the DNA, and it will start the synthesis of DNA strand by adding the nucleotides. Here we can see that these are our nucleotides, this, and they have been added along with the primers. So in this way, they are going to add in this direction, and in this case, the nucleotides, they are going to be added in this direction. So these are the nucleotides. We can see that they have been added, and as the process will go on, all the nucleotides have been bound with the DNA strand, and this single stranded DNA is converted into double stranded DNA. So in the start, we had only one molecule of DNA. Now we have 2 molecules of DNA. This cycle, it is repeated for 20 to 40 times, and after this we will have so many copies of this original DNA molecule. So here you can see that we have many copies of our desired DNA. So this is how the polymerase chain reaction helps making millions to billions copies of a specific DNA molecule. So that was our lesson on what is polymerase chain reaction or PCR. If you still have any question related to this topic, you can ask me in the comment section. I will see you in the next video.